ladies and gentlemen. Today we're going to look at some chromosomal abnormalities of humans. So this is just a single little page of notes, and basically there are some different things that cause variety of genetic diseases. Some are just problems during meiosis. One of the most common problems during meiosis, during that this is going to be during anaphase as the sister chromatids are separating, is called a non-disjunction. And this would be that when you were doing that bead lab, if the beads didn't separate half and half and you got an extra bead going one way or the other way, you would end up with a non-disjunction. And some of our most common non-disjunctions, the first is Down syndrome, trisomy 21. There are 3 21st chromosomes because one went one way and 3 went the other way, and you ended up getting fertilized, so you would have an extra 21st chromosome. Edwards syndrome is 3 number 18 chromosomes, and Patau syndrome is 3 number 13 chromosomes. We'll look at what these disorders are and what are the characteristics of them in the next video. Translocations can also happen. Here's a portion of a chromosome that got broken off and then reattached to another chromosome. Our best example here is called cru de chat, also known as cry of the cat. So this would be during that flip-flopping of crossing over, and things just did not happen properly. Some of the sex chromosomal abnormalities or anomalies would be Klinefelter syndrome, XXY, these are males. They have 47 chromosomes. XO, this is a female. She has only 45 chromosomes, so she is missing an X, known as Turner syndrome. Extra X in females, XXX, triple X, or super female syndrome, have 47 chromosomes. All 3 of these are the result of non-disjunctions. You got 1 too many or too few chromosomes. Sex linkage, things that are carried on the X chromosome typically. One example is color blindness. So this talks about different types of pigment issues. Some people are just red-green colorblind, some are purple-yellow colorblind, some are completely colorblind. Another sex-linked disorder is hemophilia. This is a blood clotting disorder. It has to do with factor VIII in your blood to allow you to clot properly. Again, we'll do details of these when we get into the big packet next time. Muscular dystrophy, there are a variety of different types. One is known as Duchenne muscular dystrophy. This is a late onset type of disorder, which will result in the atrophy and wasting away of your muscle tissue. Typically skeletal muscle starts first and eventually cardiac muscle, which would lead to an earlier than normal death. How do we diagnose these diseases or disorders? One way is through a karyotype. So how do you do this is we do something called an amniocentesis. This is taking a large needle. You're going to remove some fluid from the amniotic sac of the mom. So you would go under an ultrasound so that the clinician could be able to see where the fetus or the fetus is developing, and you're going to withdraw some of the fluid from that amniotic sac. In that fluid, you're going to have skin cells and things that have been sloughed off of that baby as they are developing. And now we can look at the chromosomes within those cells of tissue, and you can line them up and you can then see, are they matches? Do you have 2 number 1 chromosomes, 2 number 2s, 2 number 3s, 2 number 4s, and so on and so on and so on. If you get too many or too few chromosomes, you know that there's a genetic condition. Some conditions can also be checked by blood test. So we can just diagnose some genetic situations. Sickle cell anemia is one that we can look at from a blood test. And here we're looking at a homozygous recessive gene. It is a single amino acid substitution. So there is one mistake that is causing an amino acid not to be the amino acid that we're looking for. So of the 20 amino acids, it just happens to be the wrong one that is in place. This disorder used to be a huge advantage or benefit to people if you were living in a region of Africa where there was African sleeping sickness. You would get bitten by an insect, it would infect you. If you had normal red blood cells, like people not of this descent would have, you would probably get the disease and die from it pretty quickly. If you had these little sickle cells, though, you either never got the disease or you survived, and you would go on to live at that time a long and healthy life. The reality then is maybe into your 40s or 50s. For us today, that's not such a long and healthy life. So as a result, this sickle cell is no longer an advantage. So at one point it was an evolutionary genetic advantage. Now, not so much. People who have sickle cell anemia, there are treatments on the market now. Their lifespan is improving. The problem with it is these little pointed edges on the sickles of these cells, as a person ages, start to get clogged up in their capillaries, causing excruciating pain, causing other problems, eventually leading again to an earlier than normal death. Huntington's disease, we spoke about this the last video, is a deterioration of a patient's brain. The patient will slip into a decline and they will start to lose their ability to communicate. It is an autosomal dominant gene, but we also talked about the fact that even though you might have this gene, you may not express it. So a person with the gene has a 50% chance of inheriting the disease. PKU, phenylketonuria. I think most of you in your first year bio should have talked about this. Here we have a problem with an amino acid, phenylalanine. Look on some of your containers in your cupboards right now, sodas, your gum, things like Jell-O gelatin. They have phenylalanine in them. And this is a condition where babies who are born with PKU, if they consume products that have phenylalanine in them, they cannot metabolize them, and as a result, it builds up in their brain. And it will lead to irreversible mental retardation. Every baby born in the United States currently is tested for PKU at birth. They do a little blood prick test. And if you were born with PKU, your family was given a little book to explain, here's the condition, these are the things you cannot feed your child until they're somewhere in the 8 to 10-year-old range, because at that point your brain will have developed enough so that this won't be as effective. Personally, I think if I was diagnosed with, I would probably stay away from those foods for the rest of my life. Albinism, we spoke about this a little bit. Being albino means you're lacking pigment. You know that you need the color gene to express pigment, but then you also would need the genes to be albino. So we are missing the ability to produce melanin, which will give us our pigments. This is a recessive condition. Tay-Sachs disorder, again, when you covered in first year bio pretty thoroughly, it is autosomal recessive, so neither parent needs to have this disorder. They are passing along a recessive trait to their offspring, both mom and dad. Typically, these babies are born very normal, happy, healthy. They develop through a period of time. As they're developing through this period of time, they're meeting all of their milestones, and then there comes a day where they start to decline backwards. That is because of a buildup of lipid or fat in the brain that their body has the inability to metabolize. It will typically lead to an earlier than normal death in children who are born with Tay-Sachs. So how do we know if these things are part of your child or your developing fetus? We talked about amniocentesis, so it's explained a little bit here. Once the amniotic fluid is withdrawn, it can then be analyzed through a process called karyotyping. Again, I believe that many of you did this in first year bio. So hopefully that gives you a little more background, and we will go on to our big packet about human anomalies or abnormalities next time. Have a good day.